Thank you for inviting me here today. It's really an honor and a pleasure to be here. I am both at the Department of Neuroscience at UCSD, and I'm also a co-founder and serving as the CEO of SciCrunch Inc. There's a new nonprofit, which I forgot to put into my list of affiliations on this particular slide, but it is the RRIDs.org nonprofit, which Zach is also one of the board members for. So, to disclose all of our conflicts here. So what the heck is an RRID, right? I think that's kind of the first thing you might want to ask. And so this is what the anatomy of the RRID syntax looks like. It is it has a bunch of stuff associated with local identifiers, versions used of various resources, things like companies that provide these things. And then there is this RRID part, and this is, we'll spend a lot more of our time dealing with this thing. So this is a global unique persistent identifier. No, it is not a DOI, it is an RRID. And the reason it's not a DOI is because this is largely not a digital object. So this is a physical object. This is an antibody against the furin epitope of this particular protein. It has all of this information associated with it. It has actually a lot more. This is what we ask the authors to put into their papers. And it has the persistent unique identifier, which is that thing that then holds up and is associated with all of its metadata, all of the papers that use it. There are lots of RRIDs out there. There are RRIDs for mice, for cells, for antibodies, for pigs, also for pond scum. We love pond scum, but frogs and lots of biological things, and also software tools, instruments, and core facilities. So we're going to spend most of our time doing that, but I just wanted everybody to be aware that the driver for the RRID project, the reason that it is being enforced by Cell Press, it is being enforced by Nature, it is being enforced by Science, is that it is driven out of the biological space and trying to identify the things that go into the study, the kind of the resources, the reagents, the key pieces of that study that need to be identified a lot better. This is the author's workflow. So there's some journal that says, hey, author, go to the RRID portal, get your RRIDs for everything. And so the author has to go to the site. They have to go into the right portion of it. Here's an antibody. Then what they do is they put in a search term. They find their correct antibody. They can copy by, with a single click, or they can just, highlight a section of this proper citation, copy it. They paste it into their manuscript, and then magically that manuscript becomes data. So again, RRIDs are an agreement. These are an agreement between journals, primarily, and between stock centers, and of course, the bioinformaticians that run the kind of centralization process. But we at the RRID, at rrids.org, we do not hold mice. We do not know that much about mice. We do know things about some aspects of the research enterprise, but we lean on and we attract partner databases. We attract lots of partners, and those partners are the people, the real experts on things like cell lines, plasmids, biosamples, and other sorts of things that then become the experts and the databases and the providers of RRIDs for that class of things. And the journals have an important role here because they are the stick, if you will. And I know journals hate being the stick, but they are the place that asks the author to actually use these things in their papers. Many authors do not need a stick, and our understanding of that comes from the fact that in bioRxiv, there are more than 10,000 papers that have RRIDs, and there was no stick there. So we know that a lot of authors are doing this preemptively because they think it's a good idea. And we've just reached a milestone recently. There are now 1 million RRIDs in articles, and we know that because we track their usage. Okay. Now, this is what an RRID web page looks like. So this is something that you might use as an input to the study. In this case, this is a place where you can put data. So this particular RRID, and every RRID has a name. A lot of them have images, not all of them. It has the persistent identifier. It has where it is on the web primarily. It has a proper citation. It has some variety of description and other metadata, such as the key defining citation. And then it has some papers that are associated with this thing. There's lots of other information about the availability of the resource. Oftentimes when we have things that require a license, there is a license information in there. There's kind of items about record creation, updates, and then there's a lot of other alternative identifiers, such as DOI shoulders that actually direct some machine learning tools into the literature to then pull some of that information about who's citing, who's using each of the RRIDs. And of course, there are, for some of the types of RRIDs, we also have a lot of relationships. And I'll show you some of that live because I think it's better live. But in this case, the brain image library is actually used by BICCN, which is one of the big brain projects in the US. And it's also used by the Brain Initiative Cell Atlas Network. So we know that there are these relationships between this RRID and these other RRIDs. And by the way, for those of you who have heard of these things, and probably all of you have, DataCite also lists the brain image library, as does RE3 data, and of course, related to the US Brain Initiative. So doing a little bit more of a deep dive into how these kind of RRIDs are being cited. So, in a lot of the cases, the author does use an RRID. So this is great. The Brain Image Library asks authors to do this. So the authors do comply in some percentage of the cases. Sometimes authors will use an abbreviation. That, as you can imagine, is a little fraught. Some authors use a DOI. In this case, there is a brain image library data set, which has this particular DOI with this end part, but one of our robots can actually recognize the fact that this is DOI.org, and here is the DOI shoulder. And of course, there is the URL. So these are all different ways that you can refer to this particular RRID. And some of those are found by different means. So if an author used an RRID, we have a funded project that pays for about 2 to 3 students at a time to go through the scientific literature and find those. They run a little automated reader to pull all of the RRIDs in that paper into a database that then gets published out to various methods. If the author used a name, a DOI, or a URL, there is no human in the world that would actually find that, except maybe for one RRID. But these are all automated checks. Of course, automated checks are great. However, they have some issues associated with them. One of the issues is that all automated checks run on the open access subset of PubMed Central. We do not have access to the literature more broadly, not at this scale, not at this level. So when those things run, they do run about once a month, and then we can check for all sorts of things. And then you might ask, well, why isn't my thing, being detected? And the answer is probably we can't see it. We can't see the paper. So again, these things are a much deeper dive. They're much better in many ways. They pick up a broader set of the metadata that's associated uniquely with that particular RRID. But again, that broader set of metadata is only accessible from text mining available scientific literature. And it has to be typeset. So let's do a little bit more of a dive into how these RRIDs are actually referred to. So, for example, this is an RRID for the non-human primate reagent resource. This is a core that provides antibodies primarily. And they have been going through the literature and spending a vast quantity of time basically querying the literature, finding out each of the reagents that they distribute to a particular lab. They track that lab for a certain number of years, and they try to find any mention of any of their reagents. And so here's one of those papers. So there is a reagent ID in here, which is great. This is not a persistent unique identifier, so you might find this string in many other papers. The authors actually acknowledged a former core director, so this Keith Riemann. And then the author thought that the name of the resource was the National Cell Culture Center. It was never the National Cell Culture Center. So, but it's a valid way to dig into the literature, but it just takes a tremendous amount of energy to be able to do this. So we've been working with the ABRF core marketplace to improve the way that people talk about these core facilities primarily. And so the ABRF core marketplace has an RRID with a little cite this button, so you can just click that, copy it, and paste it into your paper. Each facility can also pick up that button and they can put that on their website. We have other, tool tips basically and tips for how do you get people to do this better. And we had actually asked, so back in 2019, 2020, when we started this process of registering a lot of a lot of core facilities to the RRID, working really closely with the ABRF community, and we noticed that some cores were getting a lot of citations, some cores weren't getting very many citations. So we kind of surveyed the top 25 cores, and the facility heads actually told us what they were doing, which was great. So, the top ways that the cores got compliance is that, the website of the core actually has a very clear and short, this is important, short, brief message. Here's we want to be cited. Copy this, put it in your paper. That's it. A lot of the cores that weren't so successful had like a page-long diatribe on why they should be cited, but then not an easy way to cite them. Diatribe bad. Short little thing actually turned out to be very good and effective. An email signature with where the director or whoever it is that's doing the primary communication with the people using the cores was a really good place to put an RRID because it put it got that number into everybody's subconscious when they're looking at their data or when they're getting their results or what have you. Always at the bottom there was this: using our stuff, are you going to cite us? This is how we want to be cited. And then of course there were lots of other really creative solutions, things like newsletters, pasting the RRIDs on top of the instruments, on top of like in the core facility and everything else. But those two, the website and email, seem to be really simple ways to get a pretty good amount of compliance. So you can see that RRIDs are actually increasing here. So we did this analysis last year and at the end of 2024, actually, and you can see that about 2,000 had been cited in 2024. Now you might want to say 2,000 out of what? Is it 2001? Is it something else? Well, we also were monitoring for the core facilities citations by grant number. And so looking at the grant number data, so again, we don't have grant numbers for all the cores, but for the ones that we do, we had about 2,000 citations by grant number. And now in the biomedical space, I don't know if all of you are aware how this happens. The core facility itself can do all of that monitoring the way that we saw before in my slides talking about that national primate reagent resource. They do that, and then they assign those papers into their RPPR reporting system at NIH. So the core facility owners largely have to put those things into the systems that then report on those. So, the light blue here are the grants. This is work that the core facility has to do in order to get those citations to show up. But the dark blue, the RRID, that's what the author has to do. And so the amount of work that's being done here by the facility owners is far lower. And when you're actually looking at these facilities, you're getting more and more of the citation moving towards the kind of pushing this onto the author by having some of those really clear instructions. Okay, so I do not do anything alone. This is the Fair Data Informatics Lab, or FDI Lab for short. We do have funny hats, and there's lots and lots of people involved in this effort.